Hello, guys, and welcome back to Cancer Wars Episode 3, which is Revenge of the Frequently Sacrificed Cells who got tired of the altruism and became selfish and therefore cancer cells. In other words, it's about how cancer cells evade apoptosis, which is, you can consider the cell's primary mechanism of getting rid of the useless parts of the body or a tissue that are not going to contribute to its welfare. And naturally, that's detrimental because each and every cell in the human body is dependent on the function of the other. So in the best interest of everyone, it should stay active. But cancer cells are extremely selfish. They don't rely on this. They don't believe in teamwork at all. And today we're going to talk about how they evade apoptosis. So you can consider it like this, right? Just as how cities rely on vigilant security systems to maintain order, our cells employ intricate mechanisms to ensure their health and balance. And today, that's what we're going to delve into. So just assume every cell to be like a bustling city with a security system that monitors health and population. So this system includes receptors on the cell that act as security cameras, and these receptors detect signals, some promoting cell survival like the growth factors such as IGF-1 and IGF-2, and others signaling danger such as FAS ligand and TNF-alpha. And a quick note about IGF-1 and 2, they bind to the receptors IGF-1R, and for the FAS ligand and TNF-alpha, they interact with FAS and TNF-R1 receptors respectively, which are with R meaning receptors naturally. So IGF-1 and IGF-2, which stands for insulin-like growth factors, are key survival signals that bind to IGF-1R, initiating a cascade of intracellular signaling pathways, including the P13K-AKT pathway. I'm not sure how biologists pronounce it actually, because I've just read it. I haven't really spoken to anyone about it, so I'm not sure if it's Pi3K Act or you're supposed to spell it out, but I feel like Pi3K Act would be something that's appropriate. But anyway, I'm just going to spell it out. This pathway promotes cell survival and growth by inhibiting apoptotic processes through the phosphorylation and inactivation of pro-apoptotic factors such as BAD and capsase 9. BAD, that you can say that is bad, but I feel like that can be easily confused. But pro-apoptotic factors are basically factors that promote apoptosis that are found within the mitochondria. On the other hand, death ligands such as the FAS ligand and TNF-alpha bind to the receptors FAS and TNF-R1, leading to the formation of death-inducing signaling complex, which has the name DISC, which is super cool. And this complex recruits and activates initiator capsases, such as capsase 8, which subsequently activate effector capsases and drive the cell towards apoptosis. So again, just re-emphasizing this concept that's found repeatedly. The cell is like a huge flowchart, and everything just contributes to its motion, right? Inside the cell, internal sensors detect anomalies such as DNA damage or activation of oncogenes, and the tumor suppressor protein P53 is a crucial internal sensor that responds to DNA damage by upregulating pro-apoptotic genes such as BAX, PUMA, and NOXA, which is BAX, PUMA, and NOXA, thereby promoting apoptosis. Now we're going to talk about mitochondria and the BCL2 family, who are the guardians of cellular fate. So deep within each cell, the mitochondria acts as a control center, similar to vaults housing vital resources. And when the city faces a crisis, pro-apoptotic signals converge on these mitochondria, causing them to release cytochrome C, which is like an alarm signal that initiates apoptosis. Now, the mitochondria is going to have multiple functions that you might learn about, and in not all functions does it act the same, similar to how one building in a city might be used for multiple purposes. So this is just its purpose within this large context. And it's very important to understand biology in a context, in my limited experience. So the release of cytochrome C from the mitochondria is a critical event in the apoptotic pathway. Once in the cytoplasm, cytochrome C binds to APAF1, which is apoptotic protease activating factor 1, and ATP, which forms an apoptosome. The apoptosome then recruits and activates capsase 9, which in turn activates downstream receptors such as capsase 3, 6, and 7. So the BCL2 family of proteins regulates this mitochondrial pathway, because you don't want pro-apoptotic signals all around willy-nilly, right? So pro-apoptotic members like BAX and BAC oligomerize and form pores in the mitochondrial outer membrane, facilitating the release of cytochrome C. Anti-apoptotic members like BCL2 and BCLXL prevent this process by sequestering pro-apoptotic proteins and maintaining mitochondrial integrity. The balance between these pro and anti-apoptotic proteins determines a cell's fate. fate. Always a hard word for me. I don't even know why. So now we're going to talk about capsases, who are the executors of cellular cleanup, who are the ones who do the stuff, right? So once a decision for apoptosis is made, capsases emerge as the cell's demolition crew, which is like the coolest job ever. So capsases are cysteine aspartic proteases that exist as inactive proenzymes and are activated through cleavage in response to apoptotic signals. So there are two main types of capsases: initiator capsases, for example, capsase 8 and capsase 9, and effector capsases, which are capsase 3, 6, and 7. Initiator capsases are activated in response to signals from death receptors or mitochondrial release of cytochrome C. Once activated, they cleave and activate effector capsases, which then dismantle cellular components. And also, death receptors can be considered to come from an extrinsic pathway, and the mitochondrial release of cytochrome C can be considered to be an intrinsic pathway, because one is something that's coming from outside the cell and instructs the outside the cell, and the other one is something that's coming from within it, right? So effector capsases, and back to effector capsases now, effector capsases degrade structural proteins like actin and cytokeratins, nuclear proteins like PARP, and DNases that fragment the DNA, ensuring the orderly breakdown and removal of the dying cell. So now we're going to delve a little bit into the historical perspectives and key discoveries that made this possible, right? So looking back at scientific history, early observations made by Kerr, Willie, and I'm not sure if that's Curry or Curie, but it's probably Curry, in hormone-dependent tumors, they revealed significant apoptosis following hormonal changes, highlighting apoptosis as a critical barrier against cancer progression. The discovery of the BCL2 oncogene in follicular lymphoma provided pivotal insights into how cancer cells evade apoptosis, and it shone a light on the fundamental mechanisms of cancer biology. So, and in experimental models, the absence or malfunction of key factors such as P53 or IGF2, they show the crucial roles of these genes in regulating apoptosis. Mutations in the P53 tumor suppressor gene, found in over 50% of cancers, disrupts the cell's ability to undergo apoptosis in response to stressors like DNA damage. It is obviously something that happens in cancer. Similarly, alterations in pathways like the P13K AKT or Pi3K ACT, just like the name, diminish apoptosis and enable cells to survive and proliferate unchecked. And in the mechanisms of evasion, cancer cells employ sophisticated tactics to evade apoptosis. Some tumors upregulate decoy receptors for death ligands like Fas ligand, diverting these signals away from their unintended targets and promoting cell survival. For instance, soluble Fas or DCR3 can bind to a Fas ligand and prevent it from interacting with the Fas receptor. This is a concept that's very common throughout biology. It's also used in drugs, both the therapeutic and the psychoactive type. And understanding these evasion mechanisms of apoptosis opens avenues for developing novel therapies that restore apoptotic pathways in cancer cells. So targeting therapies that reactivating P53 or inhibiting anti-apoptotic BCL2 family members, or even modulating death receptor pathways, these really hold promise in overcoming these defenses, and they give us new hope in the ongoing battle against cancer. So by understanding how cancer cells evade apoptosis, we can develop innovative therapies that might restore balance and offer new hope in the fight against cancer. So I hope that now through this you can appreciate the cornerstone of biological resistance and hopefully be encouraged in case there's anything you didn't understand or you would like to learn more about, research and investigate on how this can be used in the quest for health and longevity.